and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers at any time in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and the moderator will ask our speaker your questions. Your questions in the Q&A window will only be visible to myself, our moderator, and our speakers. So thank you for joining us today. It's now my pleasure to introduce our moderator, Daniel Strauss from Asahi Kasei Bioprocess.
Hi, my name is Daniel Strauss. I am the Director of Research and Development at Asahi Kasei Bioprocess America, so I will be moderating the show today. Before I do, I would like to give a brief introduction about Asahi Kasei Bioprocess. Many of you may be familiar with our Planova brand of virus removal filters. They're very established in the industry. But we have many more activities that we've been involved with and business units that we have now. Included in the purification unit are the Biooptimal and Mycorrhiza microfilters, which are used for both harvest of cell cultures from normal batch operations as well as perfusion operations. So we've been expanding in that market. We have a fluid management division, which manufactures large-scale chromatography skids, filtration skids, and the motive inline buffer formulation units, which can really increase the efficiency of your manufacturing footprint. We have moved into biosafety testing services through the acquisition of VirusSure, which does pathogen safety testing, as well as Bionique, which does mycoplasma testing. And finally, we recently acquired Planova, which is a CDMO, does contract development and manufacturing operations in California. So we are now much more able to basically work with you to achieve success for your company. At Asahi Kasei, our motto is pioneering biologic safety and manufacturing efficiency. We do this through many different things, especially through unrivaled product quality, innovation, and a very dependable global business unit. But what I'd like to talk really about today, and what the focus will be today, is our deep technical support and scientific knowledge sharing. So we think it is very important that our scientists work directly with our customers, both to understand their processes and optimize those processes, but also to understand how our products can fit into the processes that exist today, as well as the processes that will be coming tomorrow. And with that, our webinar today is going to focus on continuous bioprocessing. This is really a new and exciting approach to biomanufacturing that has the potential to improve product quality, decrease costs, and really provide great manufacturing efficiency and flexibility for many different processes and products. So as we hear today, or as we hear the presentations today, as Alex mentioned, please type your questions into the Q&A section, and I will try and get to those questions for the speaker to answer. We are limited on time, so if we don't get to all the questions, please realize that we will follow up by email and get back to you with the answers to your questions. So without further ado, I'd like to introduce our first speaker, which is Kathleen McLaughlin. Kathleen is currently a senior scientist in the downstream process development group in the bioprocess R&D at Merck in Kenilworth, New Jersey. She recently celebrated her 25th anniversary at Merck. Congratulations on that, Kathleen. During her time at Merck, she's contributed to discovery, early stage, and late stage development, and she supported the commercial process development of Keytruda. Her current work involves process development for various downstream purification steps for continuous manufacturing of a monoclonal antibody. All right, go ahead, Kathleen.
Hi, everyone. Hello, and thank you for attending today, and another thank you to the organizers for inviting me to give this talk. I'm going to be talking to you today about the evaluation of the Planova BioEX virus removal filter in continuous bioprocessing of a monoclonal antibody. So I'm going to talk to you about the downstream purification process development for a specific monoclonal antibody. I'm going to go over our fed batch process 2, the switch to continuous manufacturing process development for this antibody, some considerations for continuous viral filtration, some experiments that we did with different virus filters using the batch process, and how we came to select the Planova Bioprocessing virus filter for our process. And then finally, I'll show you some pilot scale data for the Planova Bioprocessing filter. So if we look at our fed batch process 2, the overview, as is typical as you move from a process 1 process to process 2, what you're looking to do is to refine your different unit operations. There's several key ones. For us, we were looking to increase our yields across these very common purification steps. And we also needed to have better drug substance stability. So one of the ways we decided to do that was we added a second polishing chromatography step. And we did that in the hopes that we were going to clear lipases and other impurities that were affecting our drug substance stability. And also as part of process 2, we were going to do some virus filter screening. And I'll get into that work on the next slides. One of the things we noticed throughout our downstream process 2 was the presence of particulates. And these particulates, as expected, were increasing our turbidity of the intermediates. And these particulates were identified as proteinaceous, and they pose a risk. They pose a risk to filter fouling during the process. They're going to affect our robustness. They can filter our columns. It's just not a good thing. So we did several studies, and it confirmed that it was due to longer hold times for some of the intermediates, higher temperatures, and the faster pumping flow rates associated with batch processing. And as we were doing this process 2 process development, Merck made the decision to switch from a fed batch process to a continuous manufacturing process for this particular antibody. Now, this was going to be the second continuous process for Merck. And so because of the tight timeline, we decided to leverage some of the process development activities that had been done for the first program, which was also a monoclonal antibody. So we didn't have to start from scratch in terms of our continuous manufacturing process development. We had some work to go on. So as we as we make the move from fed batch processing to continuous processing, there's several considerations. And this is accomplished, continuous biomanufacturing is accomplished by integrating automation into your process using a control platform. And when you think about it, downstream continuous operation is based on cycling small batch operations. And this is accomplished through the use of surge vessels. So you can see in this diagram that the green circles are surge vessels. And speed compensation in your flow rate allows you to do this modular kind of operation. But because you're always flowing, it could be slow, normal, or fast flow rate, you're continuous. So one thing we did know is that based on a fed batch process to a continuous process, there's significant differences operationally. And the main one is that there's a 10 to 20 time reduction in flow rate with a continuous operation versus a batch. And we had to assess, how is this going to affect our antibodies, specifically in relation to the particulates that we had been seeing in our batch process intermediates, and what's going to be the impact on nanofiltration or viral filtration. So as we consider doing continuous viral filtration, there's several considerations and challenges. And obviously, the higher you can load on your virus filter, the fewer changeouts you'll have. And as we look at the process as it goes continuous, you're looking at your multi-column capture for protein A, and then you're looking at flow-through chromatography for your polishing steps. And that feeds into your virus filter. And so we were looking to minimize the number of VF filter changeouts, increase our loading, maintain our flux. We don't want flux decline. We want to minimize the number of pressure interruptions from process pausing, which is related to your surge vessels. And we also want to minimize our system holdup. Then there comes the question of integrity testing of our virus filter. So to have the greatest operational flexibility, you really want to have your filter changeouts happening anywhere from every 3 to 7 days. Any more often than that is an operational burden. We also, again, like I said, want to maximize our filter loading so that we can lower our membrane area requirements, and then the process will cost less. And when you operate for a longer period of time, as you do in a continuous operation, you may encounter lower flux. And does this present an integrity challenge to the virus filter? So these are things we're going to be looking at. And so we do our virus filter screening in a batch process so that we don't have to run as long. So the next couple of slides are going to show you a series of batch virus filtration studies that we conducted. So our first experiment looked at a filter that I'm going to call competitor filter 1. We chose this filter first because the previous program that went continuous manufacturing used this filter. We had experience with it. It was a 2.8 centimeter squared filter, and we had used just the typical 0.1 micron pre-filter to filter out any particulates. Our feed was at a pH of 7.2, and our conductivity was at 15 millisiemens per centimeter. This experiment was run at constant pressure, and you can see the graph on your left-hand side of pressure versus loading. And then in the top right, you can see a graph so that we can calculate Vmax. And Vmax is the maximum volume of fluid that will pass through a filter before it is completely clogged or plugged. And it's determined by taking the inverse of the slope of a plot of time over volume versus time. And that's the graph that's shown in the top right. And it's common to use 50% of your Vmax number as the maximum loading for a given filtration step. And that generally allows you to have some robustness in the step and also for a safety consideration. So as we can see, competitor filter number 1 performed okay. We were able to calculate a 50% Vmax of 140.5 liters per meter squared, and we were able to achieve a loading of 254 liters per meter squared. Not bad, not great, but not bad. I should mention that the turbidity of this feed was only 4.5 NTU. So you'll see how that's going to play a role in the next experiments. As we look at our second virus filter study, here you can see our first comparison of the Planova BioEX compared to competitor filter 1. Again, this is at constant pressure. Our feed is again at pH 7.2. Conductivity is a little lower at 13 millisiemens per centimeter, but our turbidity is a little higher. It's at 11 NTU. And if you look at the top two lines, you can see that we're comparing the Planova BioEX virus filter to the competitive filter 1. They both have a 0.1 micron pre-filter. And what you can see is that just comparing them head to head, the Planova BioEX filter achieves higher loading before flux decline of 303 liters per meter squared compared to 170 meters squared. If we add an additional pre-filter to competitive filters number 1, the flow, you can achieve comparable throughput with that filter compared to the Planova Biorex. You get 305 liters per meter squared. So what we can conclude from this is that both filters were sensitive to the increased turbidity due to the particulates in the feed, and we were not able to calculate Vmax for those first two runs. And for the third line, when we used competitive filter 1 with 2 pre-filters, that Vmax, we were able to calculate it, but it's not a great number. It's 208. So what we can say is that we saw both pre-filter and main filter fouling. Head-to-head, the Biorex filter performed better compared to filter 1 if we just look at overall loading that was able to be achieved. And by adding an additional wider pre-filter, we can get the performance of competitive filter 1 to be comparable to the Planova Biorex with that extra pre-filter, which we would not want to do in our process. So then we did a third study. And again, we are comparing the Planova Biorex to competitive filter 1 at constant pressure. Now, in this case, the feed pH has been lowered to a pH of 6. Conductivity is similar to the prior experiments. However, our turbidity is even higher than the previous experiments. It's at 53 NTU. So when you look at those runs, we did add an additional pre-filter to the competitive filter 1 stream. And what you can see is that while we cannot get a 50% Vmax number for Planova Biorex, we do get much greater loading through that filter with compared to the competitive filter 1. So what we can see is that the Planova Biorex performs better when your feed is at a pH farther away from the isoelectric point of our antibody. And so we had done this purposely to try to move away from the PI of our antibody. And if we just compare to the last experiment, you can see that competitive filter 1, when you have the 2 pre-filters, in the last experiment, we were at 305 liters per meter squared. Now we've dropped to 163 liters per meter squared because of that increased turbidity. And our performance now on the Planova Biorex is better because we're at a pH that's lower than the PI for this antibody. Okay, so then if we just look at one more experiment run in batch process, again, constant pressure, we're at a pH of 6, which we know is favorable based on the prior experiment. Looking only at the Planova Biorex virus filter, we can see that, this is at an increased conductivity, 25 millisiemens per centimeter. There is no effect of that increased conductivity on the performance of the filter. And we're actually able to achieve greater loading at 473 liters per meter squared. And we are able to get a solid 50% Vmax number of 312 liters per meter squared. So this is great news. And we've decided that we're going to use the Planova Biorex filter in our process. So now I'm going to go into some considerations for viral filtration running continuously that we had to consider before we implemented this in our process. So we decided that we were going to use the 0.1 meter squared Planova Biorex filter. We had to consider the titer and working volume of our bioreactor. And we also had to decide what the feed pH and conductivity was going to be for our virus filter feed. Now that's determined somewhat by what's coming in from polishing chromatography step 2. So we actually decided that that product was at pH 7.2, but we know that we're going to do better if we lower the pH to pH 6. So we did take that polishing chromatography product and lower the pH down to 6. Now, in terms of the automation parameters, we have to determine how full we're going to fill our surge vessels, and we're going to have to know what those set points are for low, normal, and high. And we have to know what the target loading is going to be for the Planova Biorex filter. That's going to determine how much volume we're going to be able to process through that filter. And then that's going to determine how many filters we need for the process. And again, the flow rate has to be determined to be either low, normal, or high. And then for the flushing, I'm going to bring up the way that we set up the filter banks. We have to decide how much volume we're going to use to flush the filter. And that's going to be a pre-use and a post-flush filter flush. And that is going to affect your yields, and it's going to affect your conductivity. And so all of these things have to be considered. And finally, all of these factors will determine how much time each filter is going to be used and how long you can process, how long your process is going to take. And so this is showing you the two filter banks. And what you can see is that moving from left to right, you have either your feed or your buffer coming in to either filter bank 1 or filter bank 2, and then it can decide, you decide whether it's going to go to waste or it's going to go to your product vessel, surge vessel, and then feed into your inline diafiltration single pass TFF. One of the other things I consider that I forgot to mention was the max number of pauses you want to see each filter bank see before you switch to the other filter bank. So you always want fresh filters available in the other filter bank that's not being used. And then this is just a quick picture of what that setup actually looks like at Merck in our continuous manufacturing lab. So you can see the 0.1 meter squared Planova BioEX. You see two of them, one filter bank, second filter bank. And then on the bench, you can see our 0.1 micron pre-filter. Okay, so now to get to some pilot scale results. So again, we use the Planova BioEX 0.1 meter squared filter. We set our target loading to 300 liters per meter squared. And that was because of our 50% Vmax number that we got from one of our batch experiments that said 312 is our 50% Vmax target. So we had two filter banks in place, and we were expecting a filter bank switch from 1 to 2 and then back to 1 every 4 or 5 days at normal flow rate. So that would be normal flow rate into your feed vessel, normal flow rate out into the virus filter bank, normal going down the line. So we did set our filter pressure limit to 25 PSI. Now, the Planova BioEX filter is rated higher. It's rated to 30. However, you need to consider the tubing and the pumps that you're using in your process. Like your tubing, if your tubing can't handle that, then you need to consider that. So we were just being a little conservative. And what you'll notice is that this graph is showing pressure over the course of the run in terms of the actual dates that the run occurred. You'll see that our feed pressure was actually less than 2 PSI. So we're running at very low pressures here. Now, in this case, the downstream process only ran for 8 days. And so we actually only used one filter bank. And you say, okay, well, I thought you were going to switch at day 5. Well, it turns out that because of the way the run happened, we were in slow mode, slow flow rate throughout going into and out of this process, this step. And so we only actually used the one filter bank. But this was a success. So then we had a second pilot scale run, but we could not run that whole process continuously. So what we did was we took some material from the bioreactor. So it went through continuously through the protein A, the multi-column capture step, and went through viral inactivation and depth filtration. And so we took some of that material and then did batch processing through the other steps. And then we took the viral filtration feed material and said, okay, we're going to do one final batch comparison experiment, run at constant pressure. This is sounding familiar, I hope. Again, that pH is going to be 6, conductivity is 15. And so this is just a final comparison comparing the BioEX to competitive filter number 1, both with a 0.1 micron pre-filter. And what you can see is that we get great loading for both filters. So we get 487 and 500 respectively, but that our 50% Vmax value that we really want to be higher is pretty good with the Planova Biorex. So we're at 453 liters per meter squared for our 50% Vmax number. So this was very exciting, confirmed that we had picked the right virus filter for this process. And so let's show you the final slide that I have for the pilot scale runs. So we'll call this pilot scale results from run 3. pH 6, connectivity 15, that's normal. Now, we've now increased our target loading to 450 liters per meter squared because we have that good Vmax, 50% Vmax number from the prior experiment. And this run was a success. The upstream process ran continuously for 31 days, and the downstream purification process ran continuously for from days 12 to 31. And so what we were able to see, and you can see it here, is that the multiple filter banks were alternately used. So it went from filter bank 1 to filter bank 2, back to filter bank 1. And underneath each graph, which is again showing the pressure over the entire duration of the run, which is from days 12 to 31, we did increase our pressure limit to 30 psi. And what you can still see, though, is that the feed pressure is pretty stable along the run. It's around 2 psi. But when we switched from filter bank 1 to filter bank 2, you can see 2 pressure spikes. And these were well understood. They were operational issues. So what happened is the first one we were able to correct that issue, and the pressure dropped again. But then when the second pressure spike happened, it automatically switched back to filter bank 1, which is what you want to happen. And so those are the dates that those filter banks were used. So it was successful, and we were very happy with those results. So that's the work. I'm going to summarize here. So I provided you an overview of our fed batch process 2 for a monoclonal antibody as part of our commercial process development. Merck decided to switch to a continuous manufacturing process for this antibody. And there were several drivers for this and benefits. And the in-process particle formation that was a challenge in our batch process, some of those contributors are lessened when we run continuously. And we were able to overcome some of those challenges. We went over some special considerations for viral filtration when running continuously. We reviewed the results of several small-scale or batch VF experiments, and we chose the Planova Biorex as our virus filter because of its more robust performance compared to competitor filter number 1. We also demonstrated the performance of pilot scale VF, and I showed you the data that confirms the Planova Biorex as our virus filter of choice for this antibody. I can let you know that our continuous manufacturing development is complete for this process. And since I gave this presentation at the workshop in October, we have successfully completed 2 manufacturing scale runs at our manufacturing facility in Switzerland. So we had an engineering run in the 4th quarter of last year, and we had a GMP run in the 1st quarter of this year. And the virus filtration step using the Planova Biorex was successful for both of those batches. So we're very happy about that. Now, we're looking to put this filter to the real test and conduct viral clearance studies. Those will hopefully happen 4th quarter of this year. We do know that for the other antibody that used the Planova Biorex in a continuous manner, it did perform fine in viral clearance studies. So we are expecting that it's going to perform fine, even well, and we're excited to challenge it. So I just want to say thank you again for your time and attention. And if there's time for questions, I'm able to take them. And a big thank you to those who helped contribute to this work at Merck.
All right, Kathleen, thank you so much. That was a great presentation. We do have some time for questions, and we have a few questions that that people have sent in. If anybody still has more questions, please continue to jot them down. If we don't have time to get to all of them, we will follow up by email. But so starting off with I hope it's an easy one, although I don't know if you can answer it. Can you say whether the molecule is an IgG1 or an IgG4?
I'm not allowed to say.
Yeah, okay.
Sorry.
Oh, yeah, no problem. Did you try doing any further reduction of the pH? So you mentioned, I think, that you went down to pH 6. Did you go any farther or lower than that with pH?
We did not. We were facing some challenges in the concentration of our feed as it was coming out of the second polishing step. And so we were really trying to minimize the dilution because that's coming in at pH 7.2, and we have to dilute it with your acid to reduce that pH. The more you dilute in that step, you're not really going to dilute it coming through through the viral filter. You could, because of your post-use flush, to try to recover all of that product and maximize yield. You'll face a challenge in your single pass TFF, right? So we tried to minimize that dilution to avoid affecting, your single pass TFF.
Sure. Okay. And with I guess with other, say, things that might impact throughput, do you have any data on freeze thaw, or I guess you haven't done any virus spiking, but do you have any concerns that the virus spike might impact throughput?
That's always a concern that it will. Based on the previous studies that have been done with the Planova bioreacts, we feel like that's our best choice to have success. But yes, it is a concern. We haven't seen any increase in particulates with freeze thaw, which is interesting. It was surprising, but we also haven't conducted the formal intermediate stability study for this program either. So
okay, right.
That would be tested with that study as well.
Right. Can you comment on so you mentioned you have surge vessels throughout the process, and I saw you had a surge vessel before the VF. Are those all basically the same format, or are there particular considerations that goes into deciding which type of surge vessel, especially like before a process like the VF?
So for the most part, all of our surge vessels are the same. And I'm not sure if it's publicly available yet. I think we designed it. And essentially, I'll just say that it's almost like a cell culture vessel, where it's got the ability to have inputs that will allow for conductivity probes, pH probes, one of each. You have to be able to get the material in, you have to be able to get it out. So you have ports that go down to the bottom. You always want to add material into the surge vessel subsurface, right? If you're dripping from the top, that creates particulates. So I will tell you that. For the most part, we try to monitor pH and conductivity in all of our surge vessels. We're not able to. So I do know for one of them, it's a little larger. And so that one doesn't have the option for pH and conductivity, but we just need more material. So sometimes you can change the sizes depending on what you need.
Can you say if you have any like mixing capability within the surge vessel? Or?
We do. They all have mixing capability.
They all have mixing. Great. Okay.
Yeah, they have a motor on them.
Oh, sure. Right.
Yeah,
and then a question. All right. Can you comment about the rationale behind the impact of pH and conductivity on the performance? And do you think that'll impact LRV when you do the viral clearance studies?
So for us, the pH definitely matters for this antibody, especially as it relates to the virus filtration. And in terms of conductivity, I don't think that's going to matter so much. But I think we were definitely happier to be at 15 millisiemens versus 25. 25 millisiemens for our feed definitely gave us better performance on our polishing chromatography step 2, but we did back it off just because of that concern that is mentioned with the virus filter. And of course, that also affects your inline diafiltration and right. So all of these things matter down the line. So
yeah. And then one final question, then we're going to need to move on to the next speaker. Sure. Can you say anything about the pressure spikes that you observed? Anything about kind of what happened there and how you ended up resolving these?
So you saw the picture, right? I can maybe go back to the picture. There's a lot of tubing coming in and out of the filters and into the surge vessels and into the pumps. One of them got stuck in a drawer. Oh. So when you, okay. So once you trace, okay. So now your filter's paused because it's reached high pressure. You try to trace the lines and you see what's going on. So that's just an example of something that could happen, right? So it's definitely about line management, like your tubing management. And so once we took it out of the drawer, the pressure went down. We were able to, So that's why you want to specify the maximum number of pauses, because every time you pause, you risk that virus permeating through the filter.
Okay. So. And actually, I'm going to add on to that, since I said that's the last question, but I noticed your high pressure was set at 30 PSI, but I know the BioEX can go up to 45.
I was rated to 40. Yes, I misspoke there. I realized it later. Yeah, so because of our tubing and our, it's at 30.
It's at 30.
Our set point's at 30. I misspoke and said that was the limit. But after I said it, I realized that it was higher. Because of our tubing can handle greater than 30.
Oh, okay. Well, that would
be because we're using little pumps and they would just burst. So,
sure.
It is rated much higher.
Okay. All right. Well, thank you, Kathleen. We do have some more questions that I'm not going to be able to get to. So we will follow up with you, and hopefully we can get those questions answered for folks. So, all right, moving to the next presentation. So the next presentation will be by my colleague, Hironobu Shirataki. Shirataki-san graduated from the physics department of Hokkaido University in Japan. He got his PhD in polymer physics, actually. He's actually been active in research and development in various fields at Asahi Kasei for over 37 years, so quite a long time. But he's only worked in bioprocess for the last 13 years. So, not a short-term by any means, but he currently is a senior consultant of scientific affairs, mainly studying virus filtration and column chromatography for the protein purification and virus reduction of biopharmaceuticals. So unfortunately, it's about 3 a.m. in Tokyo right now, and Shirataki-san will not be able to join us in person, but he did make a recording of his talk. So again, please type any questions you have into the Q&A section. Since he's not with us, I will do my best to answer any questions we have about Planova filters and continuous virus filtration in general. But any specific questions about the methodology or the results in his presentation, we will follow up and have Shirataki-san actually answer those by email. So with that, I believe we can begin the second presentation.
Hello, I'm Dr. Shirataki, a senior consultant of Asahi Kasei Medical. I'm talking about the possibility of virus filter for the continuous process. Title of my presentation is Constant Flow Rate Viral Clearance Study of Planova BioX and S20N Virus Removal Filter, and implementation into an end-to-end continuous process for map purification. These are contents of my talk. At first, introduction of Planova virus removal filtration. Then, total flow-through integrated polishing onto columns connected to virus filter. And some virus clearance test data relating the continuous process are presented. So at first, introduction of Planova virus removal filtration. we developed a filter for virus reduction for the first time in the world and launched our first product as early as in 1989. In that time, robust virus reduction by filter is quite a new idea, and we had some difficulty to be accepted. However, effectiveness of virus filtration was soon accepted. Since then, major filter companies also followed the technology, while we continue to develop new products. Planova virus filter is flow fiber module, which assures a very good scalability from laboratory use to the production scale. As shown in this picture, flow fiber of Planova filter have a three-dimensional network structure consisting of void connecting by capillary. Filtration mechanism to remove virus is size exclusion. The properties of virus filters are robust virus reduction, high protein recovery, and stable filterability. The important point is all these properties are realized at the same time. The inventor of virus filter, Dr. Manabe, proposed multilayer filtration model to explain the virus filter property. Virus filter consists of many layers. Each layer has poor size distribution. By this structure, virus particles to be removed are trapped each layer, while target proteins go through without trapped. And stable filterability is realized at the same time. The recent study of numerical calculation showed that the multilayer structure is quite effective for virus filtration, and Planova filters properties are successfully explained by the multilayer model. And now, total flow-through integrated polishing on two columns connecting to virus filtration. In the conventional batch process, full tank is set between each steps. However, full tanks stop the process, and more time and footprint are needed. So we removed all the full tank from the cell culture to virus filtration step, except low pH inactivation. This slide shows the integrated continuous process setup and process diagram. In this process, MAB was produced from perfusion CHO cell culture and harvested. The feed between protein APCC capture and polishing were adjusted at low pH inactivation. Only the low pH inactivation step was performed in batch mode. So the production batch is defined as a low pH inactivation batch. As shown in the diagram, product harvest started at 12 days of cell culture. Harvest and capture by protein egg column carried out continuously without full tank. In polishing, two columns and virus filter are connected directly, and MAB polishing is practiced by total flow-through integrated process. Regeneration of protein egg column was practiced in 24-hour cycle. During the 1-hour low pH inactivation, two polishing columns were regenerated, and virus filter was replaced. Let's see the total flow-through continuous polishing process parameters. Both AX and CX columns are run in flow-through mode, with adjusting the buffer condition to remove impurities effectively. AX is mixed mode AX, and CX is modified type CX regime. Virus filter is Planova BioX. Each column volume and membrane area of BioX were decided to adjust the throughput of process MAB. Flow rate was adjusted to process a total volume of low pH inactivation batch in 24-hour cycle. Although the first batch shows a smaller throughput, because of bubble from harvested cell culture accidentally induced into the capture pond. Residence time of each column and flux of biofilter were stable, and throughput of each columns and biofilter were as planned. The map recovery are confirmed over 90% for all the batches, and the total flow through intermediate polishing works very well. Map and impurity concentrations after each step are changed like these figures. HCP reduced drastically at protein capture and polishing, and finally reduced to less than 10 ppm of MAP. DNA reduction was practiced all steps, and finally reduced to less than 1 ppb. MAP concentration increased at capture step, and decreased by buffer conditioning at each step. MAP aggregate increased at low pH inactivation, but induced drastically at polishing. These figures clearly show that the total flow-through polishing process worked quite effectively. In the continuous process run, we were most interested in the transmembrane pressure of biofilter during the filtration. Water flux of Planova BioX at 0.3 MPa is about 150 lmh, and max allowed recommended pressure of BioX is 0.343 MPa. During the filtration in total flow through polishing, transmembrane pressure of BioX in all batches were quite stable, without almost no pressure increase, for about 24 hours in the cycle. The small enough and stable TMP during filtration suggests that even at high constant flux and very long run may be possible. Now we have to confirm the virus clearance robustness of total flow through polishing process. The first test is inline spiking. This test is inline spiking with MLB and MBM to confirm the virus clearance robustness of biofilter directly connected to columns. In the total flow-through polishing setup, 10 mg per milliliter MAP solution with no virus spike was loaded to CL columns with flow rate of 0.18 ml per minute. To column output, 10% virus spike with the same MAP concentration is added at 0.02 ml per minute. Consequently, 1% virus spiked 10 ml per ml MAP solution is loaded to the biofilter at 0.2 ml per minute. For the BioX with 0.0003 square meters, it corresponds to the flux of 40 lmh. Volume of MAP solution loaded to BioX was 300 ml. It corresponds to 1000 liter per square meter. After loading the spike solution, 60 minutes process pause was practiced, then 30 milliliter of wash buffer was loaded. Test procedures are summarized here. LRB of BioX was analyzed for both flow-through fraction and flow-through with post-wash after 60 minutes process pause. TMP of BioX during the filtration is shown in this figure. Blue line is MULB spiked, orange line is NPM spiked, and gray is no virus spiked profile. As shown here, by spiking the virus, TMP of biofilter becomes much higher than no spike solution, and TMP increases as throughput increases. In the pressure profile of spiked solution, TMP dropped suddenly corresponding to the 60 minutes process pause. After the process pause, remaining map in column is washed out to virus filter, and then TMP starts to decrease as wash buffer reaching to the filter. For both MULB and NBM, LRB value by TCID50 method are obtained as under the detection limit for both flow-through fraction and total fraction with post-wash. This result also confirms that the 60-minute process pause does not affect the virus reduction robustness of BioX. For the continuous process, flux of virus filter can be very low because of long-time filtration, and virus clearance robustness at very low flux needs to be confirmed. So, virus clearance test at low flux from 1.5 to 10 LMH was performed in the batch mode. Load time is set to 24 hours as the integrated continuous process in this study. 60 minutes process pause is inserted between map loading and post-wash in order to confirm the virus clearance robustness. The test procedures are here. 1% NBM spiked map solution was pre-filtered and loaded to BioX for 24 hours, and flow-through fraction was collected. After 60 minutes process pause, a post-wash was also collected. LRB were analyzed for both flow-through fraction and total fraction. Results are shown in this table. For all the flux, very high NPM LRV greater than 5 long is confirmed, despite very low flux and 60 minutes process pause. It is actually important that BioEX showed LRV greater than 5 even for the flux of 1.5 LMH and even with process pause for the small size virus of NBM. The pressure profile at each flux is like this. The left figure is time plot, and the right figure is plotted by throughput. The higher the flux, the higher the TMP, but kept below 0.15 MPa, far from BioX max possible TMP. Virus removability of our new product, Planova S20N, compared with BioX. 2% MULB spike map solution was loaded to each filter with throughput of 1000 liter per square meter at a constant flux of 40 LMH. 60 minutes process pause was also induced between loading and post-wash. As a result of TCID50, it was confirmed that both S20N and BioX showed high virus removability of higher than LRB4 in both flow-through fraction and the total fraction after 60 minutes process pause. In the inline spiking test, 1% spike of MULB did not result in higher than LRB4, though it was below the detection limit. But this test shows an LRB higher than 4 with 2% spike. It suggests that the spike concentration is important in virus test using MULB. Finally, virus removal performance of Planova S20N and BioX at very low flux of 1.5 LMH were compared. Test was performed in batch mode. Load time was 24 hours as integrated continuous process run. 60 minutes process pause is inserted between map loading and post-wash to confirm the virus clearance robustness. Virus clearance test procedures are summarized here. 2% NPM spiked map solution was loaded to virus filter for 24 hours, and flow-through fraction was collected. After 60 minutes process pause, post-wash was also collected. LRB were analyzed for both flow-through fraction and total fraction. As a result, as well as BioX, new product Planova S20N is confirmed to show the robust virus removability of higher than LRB5, even at very low flux of 1.5 LMH with 60 minutes process pause. In particular, when comparing the LRB after process pause, S20N shows the LRB higher than 5 with under the detection limit. This may suggest that S20N has more robust virus removability than BioX at very low flux filtration. Now conclusion. MAB solution can be highly purified by the total flow-through integrated polishing process, in which two columns are directly connected to the virus filter. Even with a filtration throughput of 1000 liters per square meter and with a high constant flux, the TMP of virus filter remains small and stable, demonstrating the applicability of virus filter in the end-to-end process. The virus filter showed robust virus removability for the total flow-through integrated polishing process, while keeping the transmembrane pressure below the max allowed TMP of the filter. The flow fiber type virus filter tested in this study showed very high virus reduction, with LB higher than 5 log, even at very low flux combined with 60 minutes process pause prior to the final protein recovery buffer flash, indicating that these virus filters are highly applicable for continuous process. This study was supported by many people. I really appreciate their kind help. Thank you so much for your attention. I wish my talk is of your interest. If you have any question, please don't hesitate to ask us.
All right. So first of all, we are out of time. So any questions you have, I will follow up with making sure Shiritaki-san replies to those. Or if you have any additional questions for Kathleen, please go ahead and send those either through the question section on the screen, or you can follow up with anybody at Asahi Kasei Bioprocess. We will make sure your questions get answered. And finally, I'd like to thank you again, Kathleen and Shiritaki-san, for your wonderful presentations. I think it's great to see that continuous bioprocessing is moving forward, and it seems like you're making some great contributions and great progress to making that really happen for real. Finally, I would like to make one request of all the attendees. Well, first of all, thank you for attending. But also, we will be sending out a survey. If you can answer that, it's very helpful for us to be able to improve the content that we provide and understand your thoughts on what we should be providing on these types of webinars. So please, if you would, fill out the survey when you get it, and we'd really appreciate that. Otherwise, I'd just like to say thank you from Asahi Kasei Bioprocess, and I hope everyone has a great day.